تحياتي للجميع. We'll get started shortly. So today we'll be talking initially about the cerebellum and its circuitry and visit some of the disorders that affect the cerebellum in the first half of the meeting, and the second half will be focused on discussing emotion and how emotion is generated in the brain and the circuits that support that. We'll start in a minute or so. Meanwhile, if you have any questions, I'll be happy to answer. So here is a question. So what is the difference between positive and negative reinforcement? Is it positive when adding a stimulus and negative when removing the undesirable stimulus? مثل ما حكينا امبارح, adding and removing is not about the valence. The valence is basically, is it gain or loss? في عندي gain or loss, okay? يعني نفترض إنه it's about money مثلا. You either gain money, which is a positive reinforcer, okay? Or lose money, which is a negative reinforcer, punishment, okay? Reward or punishment. بينما the positive reinforcement, okay, is basically I either add a positive reinforcer or remove a positive reinforcer, or add a negative reinforcer or remove a negative reinforcer. It's a 2x2 design, you see? So you can either add gain or remove gain, add reward or remove reward, add punishment or remove punishment. You see the point? So that's the difference between the valence of the reinforcer and the positive and a negative or addition and subtraction reinforcement. Okay? All right. Let's get started. سؤال سريع. غطيتوا الاناتومي of the cerebellum before? Or not yet? Okay, so it will make my task hopefully easier. Okay, great. تمام. Then I can ask you questions about the anatomy then. How many parts are there for the cerebellar cortex? ولا مش دارسين المادة لسه. Who wants to tell us what are the different regions related to the cerebellar cortex? 3, exactly. What are they? Okay, let's take a look at that then. So basically, the cerebellum is one of these. So no, molecular and Purkinje and granular are the layers of the cerebellar cortex. I'm talking about the subsections of the cerebellar cortex. As you can see, we call them the cerebrocerebellum, the spinocerebellum, and the vestibulocerebellum. All right? هي أقسام الcerebellar cortex. بينما layers, طبقات cerebellar cortex is something different, which is the molecular Purkinje or Purkinje and granular. Okay. طيب ممتاز. How does the interaction of the cerebellum happens with the rest of the brain? Through what? The peduncles, exactly. Superior, middle, and inferior. 3 peduncles. And بتعرفوا بالضبط شو كل واحد من البيدانكلز هذه. Is it afferent? Is it efferent? Is it mixed? Which one of them is afferent? So afferent is the middle. Afferent معناته accessing, entering the cerebellum, مظبوط. And like inferior as well. Exactly, that inferior is mixed. And what about the efferent, the ones exiting the cerebellum? Exactly. So the efferent is the superior. You can actually read it right here. If you want to review the anatomy, this is a very nice summary of it, and you can basically take a look at it. Okay, so superior and inferior are uh, superior is majorly, almost entirely efferent. So E with an exit. Efferent is exit. Okay, بيطلع من الستراكشر لبرا. Afferent is accessing. بيدخل على الستراكشر. Okay. So that's actually how you can remember it with this mnemonic. It makes life easier. Okay, ممتاز. إذا we know the anatomy. طيب من وين بيجي الانبوت على السيربيلم؟ Mainly and majorly. Yes, inferior is mainly mixed actually. Yes, mainly mixed. طبعا there are no rules unfortunately in the brain. It's not that simple. But basically yes, يعني mainly you can say that superior is efferent and middle is afferent and inferior is mixed. Mossy and climbing fibers. من وين بيجوا mossy and climbing fibers? Why do we call them mossy fibers, by the way? شو يعني mossy? What is moss? Anybody knows? خلينا نفرجيكم صورتهم الموسي fibers so that you might guess actually. So let me see. This is the Purkinje cell. Where is the mossy fiber? I don't think I have a picture for it here. But mossy fibers, they're called like this because they look like a طحالب. طحلب بسموه moss or mossy, okay? So basically that's how it's named. So just remember that, okay? And mossy fibers من وين بيجوا يا جماعة؟ Any ideas? Climbing fibers من وين بيجوا طيب؟ Inferior olive. Exactly. So the inferior olive gives us the climbing fibers. Excitatory ولا inhibitory the climbing fibers? Let's see, maybe we can use the next 5 minutes just to review the anatomy quickly, and then we can move to the physiology components. Again, excitatory ولا inhibitory? They're excitatory. لما نشوف شيء بنسميه complex. We'll talk about what we call the complex, action potentials or complex spikes and simple spikes. And we'll see how actually the different climbing fibers interact with the Purkinje or the Purkinje cells and how they can lead to that really very beautiful activation pattern. And okay, طيب الماسي فايبرز شو الكونكتيفيتي تبعتهم؟ الماسي فايبرز بيجوا من وين؟ بيجوا من البونز mostly, okay؟ ده طب ابسي ولا contralateral بيجوا الماسي فايبرز؟ Contralateral. Let's agree on that. Okay? Mossy fibers, contralateral. All right? So remember that. Okay, very good. So I think yeah we have a clear set of understanding. طيب in general المعلومات اللي بتيجي من الابسيلاترال والكونترالاترال شو اللي بيجي ابسيلاترال على السيربيلم شو اللي بيجي كونترالاترال على السيربيلم؟ Summary of the anatomy. And then we'll move to talking about the rest of this stuff. Motor is contra, exactly. Motor related, cortex related is contra لأنه الكورتيسيز كلها بتبعت على البونس ابسيلاترال والبونس بتبعت على الكونترالاترال cerebellum. الابسيلاترال بيجي من الفيستيبولر ومن السباينال. alright. Vestibular and spinal مش انسوا inferior peduncle at this point. Let's talk general about information, okay? So information, vestibular spinal is ipsilateral. Motor and cortex related مش بس motor طبعا is contralateral via the pons. And based on that, we can actually just again it's a simplification. There are unfortunately again no rules in the brain. nuclear, like, but the vast majority of what you see there is like this. تمام ممتاز. إذا نرجع كمان مرة, let's just summarize what we said very quickly. There are three subsections of the cerebellar cortex: cerebrocerebellum, spinocerebellum, and basically what we call the vestibular cerebellum, which has a nodulus and a flocculus. These are the subregions of it. وبرضه عندنا شيء بنسميه the deep nuclei of the cerebellum, which are basically there are three actually basically nuclei or nuclear groups in this kind of regard. The first is what we call the dentate nucleus, أو عفوا خلينا نرتبهم من الميديال لللاترال: vestigial, interposed or interpositus, and dentate nucleus. زي ما انتم شايفين, vestigial is beneath what we call the vermis, which is the most medial part of the cerebellar cortex, immediately beneath it. The interposed is beneath the, the second part of the spinocerebellum, okay, the median part of the spinocerebellum. And basically, the third is called the dentate nucleus, which is beneath the cerebrocerebellum. Okay, طبعا الانput is the cerebellar cortex. The output are the deep nuclei. All right. هل معنى هذا الكلام انه بيوصلش output على الديب nuclei؟ بلى في بيوصل output. الماسي fibers they actually collateralize. They send an out like an input على الديب nuclei زي ما شفنا امبارح في الانتربوزيتس او interposed nuclei. بس بنفس الوقت they actually go and supply what we call the granule cells اللي لاحقا راح نشوف كيف عملوا collaterals تبعتهم in the T-shaped collaterals and so on and so forth. Okay. So question, do not mossy fibers contain also afferents from the spinal cord? Well, actually, again, there are always exceptions to any rule that you can apply to brain function. Basically, mossy fibers, they, as we said, they mainly originate in where? In the pons, okay? The pontine, the contralateral pontine nuclei. But I can't say no, yani, basically. It's just that's what I want to tell you. I know the anatomical aspect of the cerebellum is super complex. But we're trying to generate these simple rules for you guys just to know the general picture. Okay? So the answer is not no, but not necessarily yes either. Okay? No, there are always exceptions to the rule. All right, and we'll talk about this in detail, and we'll get to that, and you will see exactly what we'll find out. Okay, so basically هلا عرفنا الجنرال anatomical organization of the cerebellum, the different types of the gray matter, the different types of afferents and efferents. The interaction mainly is going to be with the cerebral cortex, mainly with the cerebral cortex, and basically this is the input that comes to the cortex, to the cerebellar cortex in particular, and it also comes from the red and the inferior olive, from different nuclei in the spinal cord, as you can see here. Let me just see if I can go back to that proper anatomy slide. So basically, you can see this from the inferior olive, from cuneate nucleus, from the dorsal nucleus of Clark. These are the spinal. Basically they come through the inferior as well as the middle peduncles, and then the pontine nuclei. All right. And there are some really nice summaries here in terms of the cerebellar cortex subclassifications, deep nuclei, and peduncles. You can basically see how that's organized in this kind of regard. تمام. And basically, these what we refer to as the transverse pontine fibers are the ones that actually underlie usually these mossy fibers. Okay, so remember that. Okay, إذن حكينا برضو على transfer of information in terms of the the functional anatomy aspects of it, and then as you can see there, vestibular and spinal information comes mainly through the inferior cerebellar peduncle. The middle cerebellar peduncle contains this information that comes from the cortex, and the superior peduncle is all of the fibers exiting the cerebellum and basically mostly connecting to the to the upper motor neuron or upper motor neurons, not necessarily directly, but mainly through the the ventral and the lateral components of the thalamus. All right. فأنا بنسميها closed loops, and we'll talk about these closed loops with the cortex in this kind of regard. فبالتالي يعني أي cortical area that projects كيف شفنا في البيزل ganglia corticostriatal loops في عندنا corticocerebellar loops as well in this regard. Okay, and we'll be talking about this a little bit more in detail. طب ليش هذا الكلام مهم? Why is this extremely important in the cerebellum? It's actually extremely key because what we care about in the cerebellum is online correction of movement errors. Some movement error correction. And then ultimately that's to some extent what we also what's going to be key also for the regulation of what we call coordination of movements. So think of what we discussed related to the basal ganglia كيف عندي error correction. امبارح كمان حكينا عن what is it called, the prediction error aspect of learning. كيف عندي error correction of the cognitive component. في عندنا هنا error correction of the motor component. Okay, motor error correction. And actually the cerebellum is the hub for doing that, and we'll see how the circuitry actually supports that in the next a couple of minutes. Okay, representation, we don't really find a clear homunculus in the cerebellum. في some form of a scatteredness of the representations of the different aspects of the body, the different aspects of the periphery. And actually there is these, like, there are still clusters that are separated, and you'll find representations of the same somatosensory areas or the same basically motor areas in multiple of these clusters. And actually this is very important as well for the preservation of function in this kind of regard. Okay, so based on that, as you can see in front of you as well, this actually shows you the output of the organization, the functional organization of the outputs of the cerebellum. And as you can see here, the cerebrocerebellum is mainly going to be projected to the dentate and part of the interpositus nucleus. Spinocerebellum mainly to the interpositus and fastigial nuclei. And then basically the vestibulocerebellum it actually skips the deep nuclei of the what is it called, of the cerebellum, and goes directly to the vestibular nuclei in the brainstem. Okay, and then the only cortex or part of the cerebellar cortex that skips the deep nuclei is the vestibulocerebellum. All right, تمام. So let's talk about actually the superior peduncle, superior cerebellar peduncle. And then actually what we talked about in the context of trying to understand what happens to the afferents, projections that leave the cerebellum. Okay, so we said that actually the superior peduncle is mainly basically حكينا afferent ولا afferent؟ مين ذكرني؟ superior peduncle. Efferent. It leaves the cerebellum. Okay, so basically we're talking about the major efferent structure of the peduncles in terms of the cerebellum. And as you can see, it actually leaves from the deep nuclei, not from the cortex. And then basically it goes to the VL complex, the ventrolateral complex of the thalamus. And from there back to the originating cortex that actually projected initially to the cerebellar cortex, in this case the cerebrocerebellum in particular. All right, and هنا مهم جدا نذكر dentate interpositus, dentate interposed nuclei are the ones that actually really going to play a role in this particular pathway. All right, and then ultimately some of that goes to the superior colliculus, And تذكروا امبارح شو حكينا why is the superior colliculus important from a movement perspective. superior colliculus شو بيعمل superior colliculus what does it control حكينا عنه أو لما حكينا عن basal ganglia. Exactly, coordination of eye movements and visual reflexes and saccades, and also like head and neck movements. Yeah, so basically it's extremely important, mostly eye movements here. And then ultimately, because eye movements are extremely important for the maintenance of ultimately coordination movements and also some vestibular aspects of things. Okay. So I've read that the vestigial nuclei projects to the vestibular nuclei and continue as the vestibulospinal tract. Is that wrong? Oh, it's not wrong. Okay. But basically, um, I'm not sure about the functionality of it. So if you want to discuss anatomy, yes, that's actually it's not wrong, but it's what does that mean functionally? We don't know exactly, you see. فاحنا هون الفocus تبعنا is what we call functional anatomy, you see. We're not going to be discussing all of the projections. Otherwise, we can never put these rules, general descriptions, general, box and arrow frameworks for how the cerebellum is organized. But you can find really very minute, very simple pathways there, a few number of cells. But what does that mean functionally? Actually, this is what we want to focus on in terms of the function. Okay. الإشي الميجر. And actually ليش بحكي in this kind of logic؟ لأن لما نيجي نحكي على شيء زي superior colliculus وال superior peduncle ولا middle peduncle, we're talking about 20 million fibers. So if like 10,000 pass to some place, this is a minor number of axonal fibers that actually represent what's going on. يعني تخيل 10,000 مقابل 20 مليون و10,000 مقابل 10 مليون, you see? So basically you want to focus on the 10 million, which apparently is doing much more than the 10,000 or even the 1,000. Okay, so based on that, we want to try to understand how does this work. So let's see. So this is basically about the different outputs. We describe this as well, okay, in terms of how the deep cerebellar nuclei project to the superior colliculus and how this particular organization is extremely important for the regulation of brain stem activity in terms of the reticular formation, the muscle tone, the different aspects of coordination, as well as the vestibular nuclei, and then based on that, their effect on what we call lower motor neurons in the spinal cord. Okay? تمام. Then let's focus on what actually matters here. And ultimately, what we will discuss in terms of the circuits. Mossy fibers, granular fibers or granule fibers or granule cells, and basically Purkinje cells, and then back to the output. This is our holy grail here in terms of the cerebellar circuit. This is what you see in front of you here. زي ما انتم شايفين هنا these different layers is what one of you mentioned in the beginning, the granule cell layer, the Purkinje or the Purkinje cell layer, and the molecular layer. And as you can see, the granule cell layer has the granule cell bodies. The Purkinje cell layer has the Purkinje cell bodies. But the interaction between the granule cells and the Purkinje cell populations happens in the molecular layer. All right. And the molecular layer has a lot of what we will talk about in terms of interneurons, different types of interneurons, mainly inhibitory, and they have different types of functions. So إذا شو اللي بصير عندي؟ بيجوا الماسي fibers, mainly pontine as we said. كمان مرة pontine مش معناته إنه في شيء mossy fibers from other sources, but mainly pontine, okay? And actually, these mossy fibers do two things. All right? They synapse on the granule cells, again, the input that we talked about to the cerebellar cortex, and they do a collateral as well to the deep nuclei. All right? So كمان مرة نفس النيورون synapses على granule cell and connects to the deep nucleus. في المقابل these granule cells form these extensive climbing let me not call them climbing actually a perpendicular axons that also form these T-shaped collaterals. And actually تخيلوا almost every granule cell can synapse to up to 200,000 Purkinje cells. So this kind of T-shaped collateralization of these granule cells leads to this extensive network of connections between granule cells and Purkinje cells. And then every Purkinje cell could actually receive inputs from almost 200,000 granule cells. Okay? And again, granule cells excitatory inhibitory, جماعة. Any ideas? Granule cells كمان مرة excitatory inhibitory. Excitatory. Okay? Granule cells are excitatory. Inhibitory are the Purkinje cells. Got it? تذكروا أنا لما يجي عندي input we are being activated. لما أنا أعمل activation okay من الماسي fibers this activates the granule cells and then the granule cells activate the Purkinje. Purkinje شو بتعمل؟ It's actually an inhibitory cell. لما يكون في عندي inhibitory component then what I'm looking for is modulation of function. طيب كمان مرة the climbing fiber excitatory ولا inhibitory؟ Climbing fibers from the inferior olive excitatory or inhibitory? Excitatory, exactly. Okay, ممتاز. هذا كلام حكينا في البداية. And I want you to remember, granule cells, I mean, basically almost all of the, primary, not interneuron cell types that we will encounter are excitatory, except for the Purkinje cell. Interneurons, the three types that we will talk about are inhibitory. There are excitatory interneurons, less, less frequent and less understood. Okay? And maybe we'll mention them very quickly. طيب إذا شو اللي بصير عندي هون أنا؟ What really happens here? What's really of key importance for us to understand إنه كمان مرة بيجي الانبوت من الماسي fibers. It excites granule cells. Granule cells actually, and it excites granule cells and excites deep nuclei of the cerebellum at the same time. Mention what again? Could you be more specific? I mentioned a lot of things, so help me remember. Okay, we'll wait for the subsequent question and we'll answer your question once you explain more. About what do you want me to explain further? Okay, so بناء على هذا الكلام, excitation of the granule cells, we receive the input اللي جاي من الcortex mainly, okay, as we said, and again through the cruni, through which peduncle? The input اللي جاي من mossy fibers, which peduncle? The middle cerebellar peduncle. Okay, great. السؤال هو inhibitory excitatory neurons. The cortical neurons, مسميهم principal neurons, all of them are excitatory except for the Purkinje cell, okay, and the interneurons. The three types of interneurons, I think we'll mention them as well. As you can see here, the most important, the most frequent is the basket cell. وفي عندنا كمان Golgi cell, and we have, let me remember the third one actually, the stellate cell, okay? So we have a stellate cell, Golgi cell, and the most frequent is a basket cell. The three of these are inhibitory, are GABAergic, okay? Purkinje cell is GABAergic as well, and granule cells and climbing fibers are excitatory, okay? Mossy fibers are also excitatory. So once you get an activation coming from the pontine, the contralateral pontine nucleus, then actually you get this excitation of the granule cells, and this leads to the excitation of Purkinje fibers, which project back to the deep nuclei. Then deep nuclei get excited by the granule cells and get like the excitation goes through the granule cells and receive also inhibition from the Purkinje cell, and then actually basically that allows us to see the modulation that happens. خلينا نركز هلا يا middle and inferior لانه في عندنا مثل ما اتفقنا afferent من الميدل and the inferior. بس basically احنا كمان مرة more common is common. Let's focus on the middle because it's almost exclusively afferent. Okay, بس this does not exclude the inferior. تمام. إذا تذكروا امبارح لما كنا نحكي عن ال Pavlovian conditioning and Ibling conditioning كيف بصير ال Ibling conditioning and how does that work, and then ultimately كيف بيجي عندنا الانبوت إذا بتتذكروا من ال stimulus or what we call the CS, and the CS actually how does this integration happen basically. So it sends the input to the deep nucleus as well as the cortex. The cortex interacts, the cranial cell interacts with the Purkinje cell, and the Purkinje cell starts doing error correction. Okay. Another pathway that we should consider طبعا بعد ما تصير هاي العملية and the learning happens بصير الباثوي من الماسي فايبر للجرانيول سيل للبركينيا سيل less active. بصير مباشرة من after learning happens، واللي هو كيف شو ما سميناه learning، لما يصير السي اس يعطيني سي ار. اوكي، لما conditioned response conditioned stimulus يصير يعطيني conditioned response. It goes directly from the mossy fiber to the deep nuclei and then to the output nuclei. All right. متذكرين هذا الكلام؟ Let's go back to the I think I have it open here. Let me see. There we go. Okay. So that's exactly what you will see. All right. كمان مرة ال pontine nuclei contralateral will carry the somatosensory input, which is the CS. We called it the CS. And the other aspects of things will carry the US information. All right. إذا بتتذكروا, which is either a shock or something else, or the air puff in the case of Ibling conditioning. الibling conditioning بيجي عن طريق الانفيريور olive اللي هو بتذكروا error correction, and climbing fiber هذه اللي بتيجي من الانفيريور olive also wraps around the Purkinje cell, and then ultimately based on this, so two sources of input: the CS will US, and then ultimately the UR initially, that actually this part of the pathway after learning اللي هو من cerebellar cortex becomes degraded and it becomes more of automatic. Okay, من الpontine nuclei للinterpositus with طبعا integration of the inferior olive input as well, which is the US. We don't need at this point. And so basically من الpontine nuclei directly to the CR output pathway. This is actually what happens there. إذا في عندي two things that are happening here. في عندي sensory information نرجع كمان مرة للmovement that is coming from the mossy fibers or vestibular information or proprioceptive information. وفي عندي error signal. الerror signal هاي جاي من وين؟ جاي من الانفيريور olive, again from other types of inputs. Could be cerebral, could be spinal, could be multiple sources. إذا بناء على هذا الكلام، بناء على الانتراكشن هذا اللي جاي من الكورتكس بحكي لي شو بدي أنا what do I need to do, and saying what I did caused this and this error. okay, بناء عليه بصير الانتجريشن اللي انتم شايفينه هذا. الانتجريشن هذا it's through the connection of these sensory information that are coming من الgranules من المossy fibers على the deep nuclei, and then again getting transmitted to the Purkinje cell will a climbing fiber that actually is coming from the inferior olive that is saying okay, this is the error that happened. And بناء على هذا الكلام بصير عندي activation of the Purkinje cell to modulate the output, which is the deep nuclei of the cerebellum. اوكي. مع الوقت مع الايرور كوركشن شو اللي بصير؟ البيركينيا cells actually use one of the receptors that we call AMPA, and actually there's an upregulation of AMPA when this happens مع الايرور correction. After a few trials زي ما شفنا في learning aspect of it بصير عندي down regulation للAMPA، فبصير any effect of the climbing fiber or even potentially the parallel fibers is down regulated. بصير مباشرة الانبوت من الماسي fiber على الديب nuclei is actually what is at play here. Okay, شفنا ال two stages of learning: the modulation component with the sensory input and the error correction or the error input, how that actually modulates the activity of the Purkinje cell, and that modulates the output of the deep nuclei. Okay, so could you please repeat the error correction pathway? Happily, of course. So the error correction pathway again comes from the inferior olive via what we call the climbing fibers. And as you can see, it climbs up and then wraps around the dendritic processes of the Purkinje cell. And what you will notice is كمان مرة في عندي sensory input or cortical input, whatever it is, جاي من pontine nuclei mainly, okay, massive fibers. Again, collaterals connect to the deep nuclei and to the granule cells at the same time. The connection للdeep nuclei will cause excitation of the deep nuclei. وبعمل loop مشان يعمل output، ولكن the granule cells and the actually connect to the Purkinje cell tell them hold your horses, let me modulate your function. شفتوا كيف ذكروا كيف cortical input في الستريتم والدوبامine on the neck of the spine is modulating the function. This is very similar, okay? With the error signal coming and the sensory signal coming on the Purkinje cell, integration happens. وبناء عليه نعمل modulation activity of the Purkinje cell that inhibits the deep nuclei of the cerebellum. بناء عليه بصير عندي modulation للerror بناء على الerror والsensory input. Okay? إذا كان عندي مثلا if I want to reach out to something and I came short, it increases my reach. Okay, corrects my reach. إذا كان بعيد كثير بقلله كمان شوي. إذا كان بده كمان أبعد أكثر أبعد أكثر. So you see, this is the movement or the motor error correction. Once I reach my goal كمان مرة في عندي sensory input برجع كمان مرة يحكي لي I don't need your help. The output is working and the climbing fiber says there is no error. بناء عليه بخف الactivation. So عندي down regulation of the activity of the Purkinje cell by down regulating AMPA receptors. Okay? So after learning, it skips the Purkinje cell and directly go through the deep cerebellar nuclei. Correct. Mostly, طبعا depends. في شيء like there's no all or none when it comes to the brain, except for action potentials, at least the way we understand it. طبعا there is always some activation that happens، بس احنا كمان مرة بنحاول نبسط الأمور so that we try to understand what's going on. تمام. So بناء على هذا الكلام خلينا نقسم اللي حكينا عنه into two loops. The first based on the input of the massive fibers. The first is called the cortical inhibitory loop. ليش؟ لأنه كمان مرة بيحفز الجرانيول cell اللي بتحفز البركيني fiber that actually inhibits the output. وفي عندي deep excitatory loop اللي هو directly projects to the deep, and again these are glutamatergic cells and therefore they excite the deep nuclei. So the question is, after learning AMPA is decreased? Yes, it is down regulated in where? In the Purkinje cell. ليش؟ مشان climbing cell error correction signal يخف الeffect تبعها. Okay, it decreases the effect of the error correction via the climbing cell from the inferior olive. تمام؟ Okay, وبناء على الكلام زي ما انتم شايفين it's a more like a two loop modulation of activity that comes from inhibition initially until the modulation happens and then directly goes from the deep excitatory loop to the output nuclei once things are learned. كمان مرة كيف نعرف إنه things are learned or not? Is there an error signal coming on the climbing neuron that is coming from the inferior olive or not? كمان مرة if you're not really clear about what is excitatory, what is inhibitory, this is a very nice figure that shows you كيف الجranule cells are excitatory, climbing fiber is excitatory, okay? And then based on that, you can see the mossy fiber is excitatory and the Purkinje cell is inhibitory. And then ultimately في شغلة صغيرة نسيتها إنه هون أذكرها إنه برضو climbing cells they also synapse on the deep cerebellar nuclei. بس شوفوا excitatory again. You see? رغم إنهم بيحملوا error signal, still they are actually excitatory as well to the deep nuclei. So it's quite interesting how this happens and how the pathway functions. And the three types of طبعا is this free of other types of regulation? The answer is no. في عندنا three types of you can read about it here actually. It's three types of interneurons. بالعادة when you hear interneurons in the central nervous system they're mostly inhibitory, GABAergic mostly, okay. ذكروا ستريتم شو كانت نوع الانترنيورونز لما حكينا في البيزل جانجليا. Major type of interneurons, was it inhibitory or what type was it? Cholinergic. مش لا inhibitory ولا excitatory depending on the receptor. Cholinergic interneurons. ذكروا كويس هاي الأمور. Okay? I mentioned it a lot of times. مثل ما اتفقنا احنا بتسمعوا المحاضرة you will answer the questions. سواء في الامتحان أو في غيره. So please focus on these things. الجاباergic كانوا الميديوم spiny neurons. مش الانترنيورونز. Interneurons cholinergic mainly, alright? في الستريتم. اوكي بس هون احنا بنحكي على mainly the basket cells and زي ما انتم شايفين هون basket cell is actually as you can see here it connects to the Purkinje cell both to the dendrite and to the cell body. في عندي the other type is actually called the stellate cell which receives input from parallel fibers and provides inhibitory input على ال Purkinje cell body. So كمان مرة ال parallel fibers الgranule cell activates stellate cells GABAergic to inhibit the Purkinje cell body. So شوفوا الجرانيول cell بنفسها excites Purkinje cell بينما at the same time it excites the stellate interneuron that inhibits Purkinje cell. وفي عندي آخر نوع اللي هو Golgi cells which is excited by the granule cells and projects back to the granule cell body to inhibit it. Okay, these are the different types and circuits of the interneurons that you can see. There are two other types, as you can see here, excitatory parts, excitatory interneurons. We call them brush cells, and other types of inhibitory we call them Logaro cells. In the any minor cells, we don't know their function. Okay, in the cerebellum, but they exist. So okay, so we talked basically about how this works, and ultimately, there's actually another kind of what we call molecular parcellation of the function of the cerebellum, based on a molecule that we call Zebrin-2. Okay, and basically, as you can see, there are bands of cells that either have that molecule or they don't have that molecule. And as you can see, basically, the Zebrin-2 bands, Zebrin-2 positive bands, you can see, actually one point I missed, I didn't really talk about is what's the difference in terms of the granule cell activation of the Purkinje cell and the climbing fiber activation of the Purkinje cell. الفرق مهم جدا لأنه من ناحية how does the the spike train or the action potential is generated. تخيلوا معي الجرانule cell synapses, and we said that actually it could synapse to almost 200,000 Purkinje cell, one granule cell. And Purkinje cell could synapse to up to 200,000 granule cells. بس طبيعة السينابس هذا طبعا كل النيورونز هي tonically active يعني لحالها it's firing on its own, and then actually modulation of activity happens basically in this kind of regard. That actually translates function as it comes from the input and then translates into the outputs. ولكن الجرانule cells cause what we call simple spiking pattern. Okay, it varies between 30 and 40 hertz, يعني 30 and 40 spikes per second. and ultimately it's just the regular spike shape. There is nothing special about it. بينما the climbing fibers that are coming from the inferior olive, the way they synapse is like, if you can see this multi contact point on the dendrites of the Purkinje cell. فمشان هيك لما عندي excitation of the climbing fiber, we call it a complex spike. And the complex spike is basically, let me see, do I have any? I don't think I have any basically representation of this. But the complex spike comes with a major one or two spikes, 1 or 2 hertz basically. بعدين ايش بنسميه spikelets, small amplitude spikes that follow. So basically كأنه يعني it's like a ripple effect. Okay, it's 1, 2 spikes and then small spikelets. So this complex spike is associated usually with the firing, with basically the interaction of the climbing fiber with a Purkinje cell, and simple spikes between 30 and 40 hertz, as I mentioned, is related to the activity of the granule cell with interacting with the Purkinje cell. Is that clear? Okay. So how do we superimpose this on the the Zebrin 2 plus bands? And as you can see here, you can see that actually, so Zebrin or Zebrin 2 basically is a molecule that is found in Purkinje cells. And basically, Purkinje positive cells or positive Purkinje cells show a more like less prominence of the simple spikes, less prominence of the interaction with the granule cell spikes. Okay? Especially following a complex spike. Actually, this is probably a good way of showing this, the complex spike. Yeah, right here. Okay? So basically, when you get a simple spike in Zebrin positive cell, like a complex spike, as you can see, this is the spike, the first initial second spike, and you can see the spikelets here. Okay? This is what we call a complex spike. بناء على reception of this complex spike, you see that actually there is more suppression of the granule cell input based on that, based on the climbing cell input in the Zebrin 2 positive cells, which is less like actually you see much more prominent rate of simple spike input in the Zebrin 2 negative or minus bands in the Purkinje cells. Okay, so بناء على هذا الكلام بنشوف انه these Zebrin positive bands of Purkinje cells are much more responsive to the climbing fibers. The Zebrin 2 negative bands are much more responsive to the granule cells. Okay? What does that mean functionally? It means one is more responsive to the sensory input and the other is more responsive to the error correction. وطبعا في عندنا canonical شيء بنسميه canonical models of the cerebellum that shows that there are actually specialized columns of structures along the cerebellum vertical axis that we can take a look at. So Zebrin is actually a molecule. It's a protein that is found in the what is it called in the neurons, in the Purkinje neurons, and apparently it has something to do with GABA activity. Okay, and ultimately يعني احنا بنطلع على molecular markers هون. يعني شو اللي بذكروا كيف عملنا D1 and D2 في الستريتم. We're trying to find something similar here. Using these molecular surface markers that co-localize on the Purkinje cells, can we find different functional subclasses of Purkinje cells? Okay, that's the idea. مثل ما شفنا إنه D1 is direct pathway, with D2 is indirect pathway. Apparently Zebrin 2 plus is more responsive to the climbing neuron, the climbing fiber, and Zebrin 2 minus neurons are more responsive to the granule cells from the cerebellar cortex. As simple as that. Okay? وبناء عليه في عندنا functional aspect of the activation. I hope this is clear. All right, so we talked about how this actually kind of modulates, how modulation happens in terms of the error correction and the movement. There's actually, you can see a recording of the Purkinje cell recording of the deep nuclear cell. زي ما انتم شايفين when particular movements happen, particular muscles are activated. طبعا شوفوا كيف البركيني cell at rest والdeep nuclear cell at rest, they're active, they're tonically, اسميها tonic activity. Okay? So basically based on that, we need to realize and understand how this works. So Zebrin 2 are linked to learned outputs. Zebrin 2 are the molecule, its absence or presence is related to Purkinje cell. شو حكينا قبل شوي? When you learn an output, when it's a learned output, we don't even need the Purkinje cell. لأنه مباشرة بيجي من الماسي فايبر على الديب nuclei. Okay? Very important. Think about this. So أنا ما بيهمني الكورتكس بعد ما أتعلم الموتور aspects of the response. I don't need the cerebellar cortex basically. لأنه الانبوت جاي من الماسي فايبر مباشرة على الديب nuclei, it projects out, back out. So you don't need that. You don't need the Zebrin bands. And Zebrin bands are in the Purkinje cells. Purkinje cells are cerebellar cortex, I'm sorry, cells. Okay. And then this is the whole concept of tonic activity of basically what we call Purkinje cells and deep nuclear cells. And as you can see here, the flexion of the rest is basically, you can see this kind of flexion and extension of the rest. The flexion is the bump, the red bump that you can see here. شوفوا كيف بتنزل الactivity of the Purkinje cell, and then it goes back when that goes away. All right? So apparently something is happening here. ممكن حد يجي يسألني ليش ما بصيرش excitation when this happens. Again, it depends on the movement. It depends on which which joint are we looking at, which muscle is being activated or excited or inhibited. And then ultimately you can see the same kind of reflection of what happens in the Purkinje cell, as well as the deep nuclear cell. ليش؟ لأنه deep nuclear cell is going to be activated by the mossy fiber. The Purkinje cell is going to be also activated by the activation of the granule cell. Got it? Especially if there is no need for error correction here with the climbing fiber. Okay, prion disease, you can take a look at this on your own, guys. It causes something that we call spongiform degeneration in the, I think you should cover this in pathology. And it leads to, ataxia-like and cerebellar-like symptoms that are generated as such. But how do we know that actually, so in terms of ataxia, مين بده يحكي لي شو يعني, what is ataxia? So ataxia is basically when you break a smooth movement into its less smooth, jerky components. يعني بدل ما أحرك إيدي باتجاه الشاشة بهذه الطريقة, I do it in a scattered manner, not smooth. All right? لأنه what is lost here is actually you just follow the rough steps in order to accomplish a movement, all right? Instead of doing that in a smooth manner. And that's actually one of the major functions of the cerebellum. Coordination is part of it, of course. And actually, the smoothness aspect of it is kind of this online ability to run the process, again, without the error correction. أنا عارف بهمني إني أتحرك بشكل smooth in terms of how I navigate that spatial temporal aspect of the movement. What's really key and important here is if I don't have the components of the system to support that, then this gets expressed as what we call ataxia. And ataxia, كيف بتحركوا الataxic patients? So there is loss of coordination. You can see them basically with like very sudden and fast movements. You see? كيف marching soldiers and bands, all right? So when you see an ataxic patient, you will see exactly the crude, non-smooth movement, okay? Or loss of coordination, or hypo or hypermetria. So depending on the component that is being lost here, is it the mossy fibers? Is it the granule cell? Is it the Purkinje cell? Is it the deep nuclei? Is it the cerebellar cortex? So مثل ما ممكن تتخيلوا, basically that's what happens. How did we actually find this out? And actually another aspect of it is not just the ataxia, which could be, again, you can notice this based on the peripheral function of movement, either gait or something related to that. But you can actually see this in eye movement. There's something that we call the saccadic eye movement. We talked a little bit about it in the basal ganglia. إذا بتتذكروا كيف السبstantia nigra pars reticulata sends input to the superior colliculus, and that actually increases if you lose this kind of connection by decreasing the output of the basal ganglia. You increase saccadic eye movements. شو يعني saccadic eye movements? Saccadic eye movements is basically the eye wandering. طبعا, by the way, your eyes, our eyes, move 3 times a second. You don't, you don't perceive that movement. And basically, you're trying to kind of focus the eye field on a particular target. So this happens 3 times a second. And then ultimately, so the eye is basically trying to navigate the best way to focus your fovea, your central vision, on a particular place. بناء على كده, people start doing experiments to see if I, basically try to do an error aspect of, the saccadic movement by weakening one of the eyes. So they cut a part of the tendon of the lateral rectus muscle in, let me show you the experiment, in basically the left eye of a monkey. And هذا كلام طبعا صار عند weak eye and normal eye. أثر على saccadic eye movement in the left eye. Then actually they applied a patch to the left eye. طبعا the right eye ما تأثرت لأنه saccadic movement فيها is normal. But when you put the patch on the right eye, the normal eye, في عندي مشكلة في saccadic movement في left eye. It does not really hit the target because of the peripheral problem. طبعا ما في عندي مشكلة في الcerebellum, عندي مشكلة في ال actual, output structure, which is the eye here. So what happens after a few days of wearing the patch on the normal eye? The weak eye does error correction. لأن شايفين الانبوت عم بعمل activation للinferior olive. بحكي لهم I have an error with the climbing fiber. And then this actually continues to be modulated by the Purkinje fiber projecting to the deep nuclei, and then it modulates the eye movement. طيب إذا شو عم يعملوا بعدين في the second part of the experiment? They shifted the patch back to the left eye, and as you can see, basically, or actually you can just remove the patch. The right eye now, the normal eye, is overshooting the saccades. So instead of undershooting, all right, it's overshooting the saccades. ليش؟ لأن صار عندي compensation for the left eye to work. The right eye is overcompensating at this point. Okay, this is one example. The other example is what we call the vestibulo-ocular reflex. مين بيعرف شو vestibulo-ocular reflex? Let me, act it for you guys. Does the same thing apply to lazy eye? Potentially, yes. And then ultimately, that's actually why you wear a patch on which eye? The normal or the abnormal eye? Lazy eye نحط patch على normal eye ولا abnormal eye? The normal, exactly, so that the abnormal eye catches up. And apparently, the cerebellum plays a key role in this kind of aspect, all right? And conjugate gaze, and I mean, anything that requires coordination is going to use the circuitry of the cerebellum. This online movement error correction based on sensory input. Okay? بغض النظر شو sensory input. All right? Okay, so the vestibulo-ocular reflex is basically when I do this, I can still focus my eye on you guys. كيف? Why do my eyes not move while I'm moving my head? ليش؟ لأنه the eyes are countering the movement of my head with the same exact speed. This is what we call the VOR. Okay, vestibulo-ocular reflex. And then ultimately, how can you impair this? Now I have a representation of what is in front of me. إذا بحط, lenses that either decrease the size of the environment or increase the size of the environment, and then actually see what happens in terms of see what happens if I decrease the minifying glasses or basically magnifying glasses, and I see what happens in terms of the eye and head movements, and then remove these glasses. I'll see exactly what happens in terms of over and under compensation. You see? So basically that's how you think about, basically the contribution of the cerebellum based on sensory input and error correction. All right? So that's basically so بنقيس طبعا الهيد movement with the eye movement relationship in order just to see how is that how does that work as a concept of trying to understand the role of the cerebellum in regulating movement and regulating coordination. Okay? تمام. So basically another aspect I wanted to mention here, you will hear a lot about, which is nystagmus. Okay? Nystagmus is this kind of طبعا في عندنا horizontal nystagmus, في عندنا vertical nystagmus, depending on which ocular muscles we're talking about, which involvement of which nerves that we're talking about as well. تتذكر which nerves actually modulate ocular movements? Do you remember? And vestibular information, which nerves carries that? 3, 4, 6, 8. Yep. Okay, great. So basically, based on that, we need to continue to focus on um, how is this carried out. Nystagmus is this kind of, oscillatory movement that you notice in the eye. And basically, when the eyes have difficulty fixating on things. طبعا شوفوا هناك عملنا هذه المشكلة في saccadic eye movement. This is actually the saccade. You're actually seeing the saccade now. It happens because of a peripheral problem, because we weakened one of the eyes. But so on, it happens because of another problem, maybe potentially a lesion in the cerebellar cortex or the cerebellar pathway that relates to either the granule cells or the Purkinje cells or the, like particular anatomical malformations that actually are related to that. Okay. Okay, and we'll start in the break shortly, guys. Just bear with me, and we'll finish in the next 2 to 3 minutes. All right, we're almost done. And the second part's going to be short as well, inshallah. And don't we'll not expend all of your fuel today. And so, okay, so there is genetic analysis. You can take a look at it. And basically, something else I wanted to talk to you about is called uh, again related to ataxia. Basically, the thing that you wanna think about is kind of the loss of smooth movement or goal-directed movement, undershooting or overshooting the goal, depending on, where the problem is. Is it sensory? Is it in the, basically in the connection between the sensory input and the cerebellar cortex? Or is it in the cerebellar cortex itself? Which part of the cerebellar cortex? Or is it in the output aspect of it, in the motor component of it? Okay? All right, so basically I think that summarizes it. Any questions? Okie doke. So let's take a 10-minute break. Let's come back here at 3:15, and then we'll discuss emotions, or emotion as people refer to it, and we'll set you free after that. Talk to you in 10 minutes. 3:15. Okay, guys. So let's go back and get back to talking about emotion and see what we can do in this regard. سجا بتسأل بالنسبة للكتاب Principles of Neural Science, fifth or sixth edition. Use the PDFs that I shared with you guys. اقرأوا من ال PDFs اللي موجودين في الفولدر اللي مفروض جيهان بعتت لكم إياهم ولا ما بعتت لكم إياهم. Okay, so you should see the PDFs. ايش بنسميه basic readings. Okay, so take a look at the basic readings and just read the chapters that are there. Um, if I remember correctly, the Principles of Neuroscience is actually the fifth edition there. So you can use the fifth edition. It doesn't really, it's not going to change drastically there. I like that organization better of the book. كمان مرة المعلومات, it's not going to be really a huge update between the fifth and the sixth. And we'll take it from there. Okay. الأشياء اللي بدي أعطيكم فيها updates حطيت لكم إياها في الادditional readings. بس كمان مرة الأشياء البيسك اللي لازم تعرفوها أنتم as medical students, it should not really affect what we're talking about here. Okay. الريكوردز لحد الآن جيهان بطلب منها تبعت لي مشان أنزلهم وأحط لكم إياهم في نفس الفولدر. بس لحد الآن ما بتبعت لي اللينك بس بدون I am unable to download them. So once I get to download them, I'll put them in the same folder for you guys. But let's see how this unfolds. I don't know exactly how things will happen. بس I think يعني في أسوأ الأحوال جيهان راح تبعت لكم اللينكس تبعت الريكوردينجز. So you should have this as well. But I'll check with her today, inshallah. All right. Any other questions about the cerebellum in particular? ولا نحكي عن الإيموشن حاليا؟ Okay. What is emotion? شو هي المشاعر? Who wants to tell us? Maybe just for مشان نستغل الوقت نخلص بسرعة إذا بتحبوا. Just ask to be unmuted so that we can discuss this very very effectively. المشاعر هي الأحاسيس. Very informative يعني absolutely يعني اسمعوش اللي بقول لكم فسر الماء بعد الجهد بالماء. Who wants to physiologically for me define emotions? شو هي المشاعر? How does it form? Any ideas? When you feel afraid, when you feel happy, it's what defines or specifies the action you take. No, emotions are not related to actions. Absolutely not related to actions. They precede actions. Okay? Potentially. But not necessarily. It's not action is not a component of emotion. Physiological response to our inner feeling. Fair to say that. بس برضو العكس, هل العكس صحيح ولا لا؟ يعني لو كان عندي أنا physiological response بصير بناء عليه inner feeling ولا لا؟ If I say I want to take this pen and put it like this, and I'm activating my facial muscles of smiling, does this make me happy? Well, I should feel internally happy and then I'll smile based on that. How does it work? Something we feel, subjective experience in a specific physiological state, which may lead to a specific behavior. Again, fair, but is the opposite true as well ولا لا? And مسؤول عنهم limbic system, صحيح. But we do some action depending on our emotions. That's a possibility, but not necessary. No, it's not a necessity. Okay, ما في causal link between the two. and affected by memories. They could be affected by memories, but not necessarily, لأنه الطفل الصغير اللي has basically very limited memory can still express emotions, right? Mental expression, possibly. على كل. Um, it's there. This is a situation where there's nothing right or wrong about it. Okay, so I'm not asking you for a specific answer here because I want to show you that you know or don't know. It's not that. It's more of um, you know when you study a concept, you need to think about it. You need to think about what that means to you, what that means to you, to your experiences, to properly connect to it and properly understand it and internalize it. Okay, so that's actually really key when you study new concepts. And ultimately also very important for ايش بنسميه philosophy of science, which is apparently disappearing from our universities and our educational systems للأسف, which is basically trying to understand a little bit deeper with these things and clearer. So clarity and depth in terms of what happens. على كل a wide variety of stimuli in a context. Emotions are not stimuli. Okay, so I watched videos saying that if you smile, even when you're not happy, you'll start feeling happier. There's actually evidence toward that. There is evidence toward that. And it's not only in humans, and I spent 10 years of my life studying fear in rats. I can tell you that as well. So basically, but it depends. Is it subjective or objective? I'm talking about objective fear here, not subjective fear. Then apparently I cannot ask the rat if they're afraid or not, okay? If you can, please teach me. All right. So when we talk about emotion, it's basically a triad of things. It's a behavior. Actually, forget about that. It's not a behavior. It's actually an internal state, okay? That is composed of three components: a subjective feeling, a physiological response, and a behavior. طب شو الفرق بين physiological response والbehavior? Physiological response is something that relates to, you know, um, you know, aspects that are not expressed in a gross manner, okay? يعني if I smile, that's a behavior, all right? Physiological response, مثلا there is an increase in the production of cortisol. There is an increase or a decrease in skin conductance, what we call galvanic skin response, all right? Um, heart rate, blood pressure, respiration, thank you, exactly, physiology. So these are physiological metrics or markers. بينما behavior is like usually related to moving muscles, okay? That's actually what we usually term behavior. It's like an output. What is the output of things? All right? اللي حكى انه ممكن يكون في behavior well actually and actions, it's not necessarily actions. يعني when you smile, do you choose to smile or actually not choose to smile in response to humor مثلا? It's more of a reflex, you see? That's actually how I would like to think about it. It's not necessarily conscious behavior, okay? Not necessarily conscious behavior. So that being said, بناء على هذا conceptual understanding of emotion, we know that actually there are multiple systems that are related to emotion in the brain. The ones that regulate the subjective aspect of the feeling, the thought, the memory, the internal state that we call feeling. And actually this is where the whole study of emotion started. Then the physiology, اللي هي الماركers. مين منكم actually معه Apple Watch or any other smart watch تقدر تقيسوا عليها level of stress مثلا, this is one of the functions. طبعا the stress اللي بتقيسوا على Apple Watch is not actual mental stress. It's what we call stress volume. It's based on a function that we call HRV, heart rate variability, which relates to physical stress as opposed to mental stress. All right, so you should know that. بس في بعض other watches, they measure GSR, skin conductance. تعرف الأورا ring مثلا, basically one of the rings that are smart rings that actually you can look at and study as well. And based on that, we think about these kind of physiological markers as ways of inferring subjective emotions. Even the emotion, the feeling, is the central part of it. هل أنا سعيد؟ هل أنا حزين؟ هل أنا متحمس؟ هل أنا basically what else? هل أنا خايف؟ Okay, أو stressed. So that actually is a very important categorization of what we call emotion, which is if I take these three systems and try to categorize emotion based on the subjective feeling component as well as interaction with the physiology and behavior, بيطلع عندي two axes. واحد بنسميه arousal level. arousal بيعتمد على أي system في الدماغ. Who wants to tell me? Fight or flight? Arousal system is controlled by which system in the brain? Arousal is controlled by which system in the brain? So sympathetic, parasympathetic, autonomic, okay? So fight or flight is sympathetic, more relaxed is parasympathetic, okay? And then ultimately valence is the positive or negative aspect of it. شو مثل ما شفنا valence of the reinforcer, positive reinforcement and negative reinforcement, a reward or punishment. فعندي positive emotions اللي هم happiness and excitement. ليش بحطهم على طرفين؟ لأن واحد منهم low arousal اللي هو happiness، واحد منهم high arousal اللي هو excitement. Negative emotions are basically sadness, which is low arousal, and stress, fear, anxiety, which is high arousal. All right? شايفين هذا الموديل اللي فيه four quadrants? Valence and arousal is basically what we call the circumplex model of emotion. All right? So based on that, we try to understand emotions based on this categorization. طبعا, again, it's an oversimplification of emotions. There are some other studies that actually categorize up to 50 plus emotions based on machine learning aspects. If you're interested, let me know. I'll be more than happy to share that with you guys. Maybe I can even put it in the additional readings if you're interested to explore more. So basically, that's how we define emotions in this kind of context. طيب, how does that happen in the brain? لما نحكي على physiology component. إذا في عندي أنا significant integration of the autonomic nervous system اللي له علاقة في الأراوزل. في عندي significant integration of measurement of stimuli, positive and negative, inside the brain or the outside. So how can we integrate this? We need access to both of these systems. The ones that actually measure positive and negative valence, and the other that measures arousal. And where did the actual initial evidence for this came from? Or where did it come from? I'm sorry. It came from studies of a neuroscientist called Hess. He used actually he experimented with cats. So he would open a window in the cat's skull and do a cut alongside a particular plane in the cat brain and ultimately see what happens behaviorally. So when the cut was يعني he did something that is called decortication. كأنه بشيل الcortex كلها. All right? They cut through the cortex. And ultimately every time he did this to a cat with a particular cut, كان يصير عنده شيء بسموه sham rage. The cat would wake up just going crazy, basically hissing and attacking and until he puts it down. Unless the cut actually was after a particular landmark. شو هي landmark? The hypothalamus. So if the decortication cuts, basically the hypothalamus is basically rostral to it, then there's no rage. Hess proposed that actually apparently the hypothalamus is really key for the expression of emotion or emotional regulation because we're severing that link. هلا احنا بنعرف انه it's not necessarily the hypothalamus, it's a connection of the hypothalamus to the reticular formation in particular or to the midbrain. That is actually really key. So it's not the structure itself, it's the connection of the structure. Okay? So بناء على هذا الكلام, we now know, based on Hess's experiments, that actually the brain can regulate emotion in this particular way, and there's this localization of the regulation. طيب. So إذا في عندي هاي الconnection اللي جاي من these particular areas of the brain that we call the limbic system, and we'll talk about this in a second, that regulates something that is descending. And initially حكينا كثير عن الموtor regulation that comes from the brain. طيب, هل emotional expression regulates motor function? And how? Okay? So الزملاء اللي كانوا حدا من الزملاء كان يحكي عن الأكشن. Let's take a look at it here, okay? So you'll see that it's not called, it's a consequence. It's not causality, okay? It's not necessarily causality. Okay, بناء على هذا الكلام اجى واحد اسمه دوشين. سمعتوا فيه سابقا دوشين؟ في اسمه مرض كبير لازم تعرفوه. Muscular dystrophy, exactly the same neurologist, the same French neurologist who characterized the muscular dystrophy. He did another set of experiments, and he was actually um, he would excite the face to cause particular facial expressions by Faradization, by putting these electrodes on the face, and basically to mimic expressions of particular emotions. Okay, and he would compare the expression using the motor component of things, which is the Faradization, as opposed to the expression related to the spontaneous, natural occurrence of this particular aspect of the emotion. And he did the studies particularly in people with some brain damage. Okay? ولكن على كل خلينا so thanks to Duchenne we know that actually now there is a difference between what we call facial motor paresis and emotional motor paresis. As the name implicates, somebody with facial nerve problems, if they try to consciously, voluntarily smile, they will have an asymmetric smile, as you can see here, okay, in this picture. So in this picture, الشخص عنده facial paresis, motor facial paresis, okay? They try to smile, they have an uneven smile. But if they tell them a joke, they can perfectly smile. في المقابل, if somebody has an emotional motor paresis, a disorder or a problem in the emotional component of the circuit, if you tell them a joke, they will show you an imbalanced smile. But if you ask them to make a smile, then they should smile normally. إذا في two components of the regulation of these outputs, of the regulations of these behaviors. One is emotional or emotion regulated, and the other is motor regulated. And this particular pathway is what is called, the Duchenne pathway, the Duchenne smile. And this is what happens if you have emotional paresis, emotional facial paresis, you'll have a Duchenne smile. If you have a motor facial paresis, you'll have a pyramidal smile, اللي هي uneven smile. This basically uneven smile that you saw earlier here. Okay, and this pathway is what really plays a role in this. Okay, so في عندي an emotional expression that happens, and this emotional expression goes through the extrapyramidal projections, limbic circuits, limbic circuitry, and basically to the ventral and the medial forebrain. It activates both the reticular formation, which is what Hess identified earlier, as well as the the autonomic nervous system. All right. إذا صار عندي أنا complete control. طبعا the limbic system also receives a lot of sensory input. So we have both the valence as well as the arousal components of the emotion. مقابل voluntary volitional movement, which basically has also a medial and a lateral, pyramidal mostly. طبعا في عندنا extra pyramidal component للmovement زي ما حكينا في المحاضرات السابقة. And both of them شايفين في balanced activation of the reticular formation as well as the autonomic and other autonomic motor neurons and other motor neurons down the spinal cord, okay, and other systems in the brain. So basically, is motor activity underlying emotion cause or effect? Okay, we talked about the possibility that if I do a motor action, I can induce a feeling, or if I have an internal feeling, I can induce a motor action, which is what we call the behavior. All right. بس كمان مرة it's more of a reflex as opposed to a conscious behavior or a voluntary behavior, as you saw with what we talked about in terms of the Duchenne smile. Okay? طبعا في evidence indicates that actually the major source of emotion is peripheral to central as opposed to central to peripheral. Okay? يعني in other words, if you feel anxious, it's mostly because of a peripheral thing that is happening as opposed to a central thing that is happening. ممكن تكونوا شاربين قهوة كثير مثلا. ممكن تكونوا basically you just worked out in the gym and then actually you're so tired, you're too tired, your muscles are not feeling okay. ممكن you have pain or injury and so on. Okay? So that's how you that's actually kind of the way that we try to understand the effects of the periphery or the effects of activation of the muscles in particular and other parts of the body on the expression of emotions. طيب نيجي للlimbic system. So from the experiments of Hess and others, of course, we know that the limbic system is responsible for the regulation of emotions. طبعا when it happens in the periphery, it needs to be projected back centrally in order to be translated into what we call emotion. All right? Without the central component, you will not feel the subjective aspect of it. You not maybe the physiological component would happen, but partially, and um, and then actually the behavior might not necessarily happen as well. All right. So, or it could start from the behavior as well if you want to just go against that. That's actually also possible. All right. طيب بناء على هذا الكلام في عندنا this kind of central lobe in the brain that we call the limbic lobe. Okay. That is composed of the cingulate gyrus, anterior and posterior, the orbitofrontal cortex, basically the fornix, the parahippocampal gyrus, and last but not least, of course, there's mammillary bodies, and last but not least, the amygdala. And I can tell you a lot about the amygdala. I did a lot of my work personally. I'm an amygdala neurophysiologist. I did a lot of work in the amygdala, and a lot of the work in the central thalamus as well, related to this kind of work, to study how fear is generated in the rat and how do rats approach or avoid, it's in a naturalistic environment. So in this kind of regard, these are very well interconnected. طبعا one most important thing as well, the ventral striatum, nucleus accumbens, is also another really important component, and dopamine, of course, and serotonin of the limbic pathways. Okay. You can study about the Pabst circuit if you're interested. This is more of like a historical aspect of it. It's up to you. So the amygdala اللي معناها بالعربي اللوزة, okay? يعني amygdala in Greek means اللوزة, okay? Because it looks like an almond. In the in the medial part of the so medial temporal lobe aspect of the brain, and it's actually composed of multiple nuclei. The basolateral is the major input nucleus of the amygdala, and you have the central nucleus. And basically, the central nucleus is divided into central medial and central lateral. And actually, mostly the output, the central nucleus is the output structure of the amygdala. The amygdala is so well connected with the cortex, reciprocal connections, especially what we call the medial prefrontal cortex and the orbitofrontal cortex, and actually roughly translates in the animal to infralimbic and prelimbic cortices, as well as the the orbital field as well in the rat in particular. It's also very well connected to the basal ganglia in the ventral striatum in particular, the nucleus accumbens, and it projects heavily to these particular areas, and it's also connected to what we call the mediodorsal nucleus of the thalamus. طبعا other thalamic nuclei heavily project to the amygdala, in particular the what we call paraventricular nucleus, as well as the central median nucleus of the thalamus, the core, the very center of the thalamus, is the heaviest projection. طيب, what's the largest input to the amygdala? المفروض أخذتوه في الأناتومي هذا الكلام. من وين بيجي؟ You should know this stuff. in the anatomy ولا ما خدتوهوش لسه؟ Any ideas? So it comes from the insula, okay? Insular cortex, anterior insula in particular. تمام. Why is the amygdala important? The amygdala is important because it's extremely key for what we call fear acquisition, fear learning and extinction, okay? In particular, fear learning, the acquisition of what we talked about yesterday, which is the CS-US UR-CR relationship. And how does that happen? So basically, when we have a tone that we're trying to pair to a shock in order to produce freezing, CS, US, and again, response, UR and then CR. How does that happen? The amygdala is really central to that. And if you lesion the amygdala, this kind of learning does not happen. It does not take place. So based on that, what you would expect, a tone is gonna come through the auditory pathways to which part of the thalamus processes auditory information? يلا يا جماعة. Ventromedial? No, not ventromedial. Medial geniculate, exactly. A lateral geniculate processes what? Vision, exactly, exactly. Medial is auditory and lateral is vision. All right. سؤال ثاني, would there be hypertrophy of the amygdala? We don't call it hypertrophy, we call it increase in volume. Okay, it's unlike a muscle. There are some reports that actually suggest that there is an increase in the size or the volume of the amygdala for for there is a difference between males and females, but like some people reported that, although recent studies suggest that actually does not exist. The connectivity, actually there are some differences between males and females. For example, exposure to fear, انه هاي فيها بصير عندي excessive support of the amygdala neurons. What actually you see is in PTSD, for example, we'll talk about PTSD and depression. You might see actually larger size amygdala. Unfortunately, meta-analysis did not really confirm that. Okay, that size actually matters in this kind of context. لأن كمان physiologically size does not matter really. بس بتشوفوا هذا الفرق في السايز when it comes to another structure. ممكن كمان مرة تحكوا مع الدكتور حاتم عن الموضوع لما يعطيكم الميموري, which is related to what is it called, location memory and spatial navigation. ممكن تقرأوها في شيء اسمه London Taxi Driver Studies. And actually فرجوا كيف the size of the hippocampus in London taxi drivers after their 4 years or 5 years of training became larger. In the amygdala, the evidence does not necessarily hold. Okay, there. بس في طبعا sex differences in this kind of regard. Okay, doke. So basically, as you can see here, we get this kind of auditory input from the CS, goes to the medial geniculate, and then directly to the auditory cortex, and at the same time to the BLA, to the amygdala, basolateral amygdala, to be processed. And then, شايفين, it's automatic. It's not, so it goes to be interpreted in the auditory cortex, and at the same time to the fear center, to the amygdala. And then if it's a fear stimulus, if it's followed by shock, which comes from other projections, بتتذكر حكينا عن inferior olive and the red nucleus and so on and so forth, or the other somatosensory aspects peripherally, if it gets integrated to something, then the amygdala is the one that acts. نستناش الريسبونس تبع الكورتكس, لأن الcortex is slow to respond. The amygdala is automatic. It immediately goes to the output circuits that regulate, that actually lead to what we call the fear reflex, withdrawal or escape. حكينا عنه مطولا امبارح, freezing. So you freeze, you flee, and then you attack. These are the different phases of the fear response. Okay? So based on that, we try to understand how and why the amygdala is extremely important when it comes to expression of emotions. طيب ممكن حد يجي يحكي لي, طب فهمنا هذا الفير. What about other emotions? The short answer to this is that fear is the most well-studied emotion. That's why mostly this is what you will hear about when you talk emotions. Unfortunately, the rest are not well studied. Okay, and the reason behind that is we could not really translate the subjective experience to proper objective and neural circuitry correlates in order to properly study them. But with the introduction of what we call functional MRI, there is much more research, but mostly in the social neuroscience aspect, as opposed to the neurophysiology component of it. Um, in humans, as you can see, these are patients with, uh, um, um, complete loss of the amygdala bilaterally. And as you can see, the person mainly had problems experiencing fear as an emotion, as opposed to other controls with other types of brain damage. All right. They also had a surprise مثلا. It's, as you can see, the green one is less pronounced. As you can see, the anger one is less pronounced, but the fear is almost completely abolished. So it's almost specific to the fear. Doesn't necessarily mean that it's very specific to the fear, but there is some specificity there. Okay. And you can actually study this even further with the identification of how do you study this? Basically, by the identification of the emotion in different, pictures that we look at and see in this kind of regard. Okay, so we have like 15 minutes left. We'll not use them all, but I want to walk you through some of the aspects related to affective disorders, depression and in particular. Okay, so affective disorders or mood disorders. Depression is actually the perfect example. And we know that actually there are significant differences between patients of depression and non and patients who don't have depression, especially when it comes to the activity of the amygdala. All right. So it's basically when you look at this kind of expression of unipolar, what we call unipolar depression, you can see that actually there is an increase in activity in the amygdala. As opposed للزميل أو الزميل اللي سأل عن increase in size. No, not necessarily size, but increase in processing. This is actually what we call the affective processing of stimuli. يعني كأنه كل stimuli بتصير كأنها لها علاقة بالفير أو السترess أو الانxiety or something like this, or potentially to the mood component in the case of affective disorders. Okay, so this is what we see. So the amygdala apparently is quite involved, and this is the medial and the orbital frontal cortices. As you can see, this is part of the cingulate cortex. Again, the limbic system and the mediodorsal thalamus. That's what they can see here. So parts of the limbic system are either excessively activated or more inhibited in these patients of depression. Okay, and actually that actually could modulate how patients respond to treatment. And I can tell you much more about this if you're interested. But maybe you can reach out and I'll send you some reading material on that. That's a major part of what I did during my career as well. طيب في عندنا cortical lateralization of expression of emotions. We'll talk about this on in the language lecture. And basically شو بسموه prosody, or if you lose it, it's called aprosody, okay, or aprosodia. And that's actually mostly localized to the right hemisphere. And then ultimately, basically, there is also an asymmetry or basically a lateralization of the processing of emotional information, that actually the right hemisphere is involved in more negative emotions, while the left hemisphere is more involved in positive emotions. Okay, again at the valence level of things. Arousal اتفقنا إنه sympathetic parasympathetic, there you don't see a lot of lateralization there. And actually, case in point here, you can see this kind of asymmetry in smiles. لأنه زي ما انتم بتشوفوا هنا, when you ask somebody to smile, they could be happy and maybe they could be smiling and sad at the same time. And this kind of fight between the motor, the volitional component, as well as the emotional component, would lead to these asymmetries in the expression of these facial expressions. Okay, this is about trustworthiness as well. You can take a look at it. And then ultimately, last but not least, is related to addiction. And actually, this is a slide I was looking for امبارح لما كنا نحكي على addiction. How you can see the effects of basically. Okay, question. So if the right limbic system is lesioned and the left will overcome it, will it be more positive? Well, not necessarily. So not necessarily. كمان مرة it's not just a very well localized um, function. يعني you see more expression or less expression, but not cessation of expression, you see? So it's not all or none كمان مرة. لأنه the brain is so plastic, it could actually account for some amazing effects or amazing functionalities even with half a brain. Okay? So this is something that you will see as you study the brain better if you're interested more in the brain or how the brain works. And بس خليني هنا let me focus a little bit on addiction. Why do we care about addiction and basically the limbic system? تذكر هون بس a big quick review of the limbic loop, corticostriatal loop في البيزل ganglia حكينا عنه, where actually it starts in the cortex, orbitofrontal cortex, and a cingulate cortex, temporal cortex, and then goes to the ventral striatum, and then from there طبعا الفنترال striatum في heavy projections من الفنترال striatum من الاميجدلا to the ventral striatum ومن الاميجدلا لكل الكورتيسيز هذه, all right? Then it goes to the ventral pallidum, basically when we're talking about basically the globus pallidus internal in this kind of case, substantia nigra, pars reticulata, and then the medial dorsal nucleus of the thalamus and back to the cortex, and even sometimes back to the amygdala, all right? So in this kind of regard. So ذكروا هذه loop, corticostriatal loop, that we called it the limbic loop, okay? And ultimately, why is this important? Because this limbic loop has a lot of dopamine in it. And addiction, a lot of these addiction drugs are related to dopamine, such as cocaine and amphetamines. They increase the release of dopamine. Alcohol, opiates, nicotine, basically cannabinoids, many different types of them. They're related to either dopamine or medium spiny neurons in the ventral striatum in particular, because these are the ones that are linked and connected to the limbic system. And this is basically, you see that some differences in terms of the expression of some molecular markers. If you're interested in them, you can look at them. In particular, you can look at this Krebs marker. FOS is basically a marker that relates to the activation of cells, but Krebs is related to the activation of nuclear activation of cells as well. So these are things that you can actually take a look at. I'll leave this for the lecture that we talk about where we talk about advanced decision making, but it's related to dopamine. كمان مرة هذا Pavlovian conditioning. Reward leads to what we call a prediction error signal in dopamine. Okay. So there's a question. It increases the wanting but not the liking. Now these are things that actually, yes, so increase the wanting but not the liking. But if you increase the wanting and the wanting happens, then subsequently you will increase the liking. You see? لأن هذول مش بس dopamine. When you say cocaine, it also increases the internal internal opioids and the other hormones and other neurotransmitters in the brain that increase the liking as well. Okay, so it's not just dopamine, but dopamine is one of the major modus operandi that we're going to be looking at, the ways the mechanisms of action. So dopamine is related to what we call prediction error. The larger the prediction error, the larger the signal that we call phasic signal of dopamine. مثل ما شفتوا سابقا نحط conditioned stimulus that is related to the US, which is reward. بصير الدوبامين neuron firing to the conditioned stimulus. بطل يهمه reward, صار يهمه stimulus that predicts the reward. Okay? And if you actually present the conditioned stimulus and take the reward away, you'll see a dip in the activity. كمان مرة negative prediction error, because you gave them the CS without the US. All right? So we'll talk about this more tomorrow. عفوا يوم I think السبت لما نحكي عن what is it called عن complex decision making and planning and so on and so forth. Last but not least is post-traumatic stress disorder. طبعا probably hear this a lot these days in the context of the Gaza war and what happens in Gaza. Psychological trauma leads to different expressions of symptoms. hyperarousal, re-experiencing of trauma symptoms, and hypervigilance. So basically, um, and avoidance, of course, so the three categories: arousal, avoidance, and re-experiencing. These are the three core components, core symptoms of PTSD. And recently, في DSM-5, they added anhedonia, loss of pleasure, to it as well. So زي ما انتم شايفين, basically avoidance is related to negative emotions as well. Re-experiencing is also another thing related to negative emotions. And arousal is this kind of startle response, also related to heightened sympathetic activity. So it's all about the emotional expression in this kind of context, in this regard. One of the pioneers in this field is Mohammed Milad, as you can see here. Palestinian من Gaza. He was at Harvard Medical School. He was one of our collaborators in the Palestinian Neuroscience Initiative, currently at NYU. He studied this in PTSD and non-PTSD patients using the fear learning paradigm, fear learning and extinction. وفرجة إنه basically what's extremely important is this kind of difference in activity between PTSD and controls in particular areas of the ventromedial prefrontal cortex as well as the anterior cingulate cortex that increase or decrease extinction of fear memories. All right, not erasure, extinction. ذكروا extinction. CS, US, stimulus A, often context A, CS without the US in context B. If you fail to extinguish, معناته the CS will always elicit the CR, okay, with or without the US in both of the contexts. Okay, Jama'ah, so this concludes our discussion of emotion. Any questions? All right, so الله يعافيكم. I see that physiology. خليني أبلش في السؤال الأول. People asking the PNI, about the PNI. I'll tell you a little bit about it هلا إن شاء الله. Are we required to study every detail in the book you listed? Should we just read it and memorize what you focus on and during the lecture? كمان مرة, I think this is the fifth or sixth time we repeat this. مطلوب منكم اللي بحكيه. Okay? اللي بده ينجح يسمع المحاضرة. Please focus on that. Okay? Take it easy, take it simple, focus on the concepts. الهدف مش تحفظوا الديتيلز، تحفظوا الديتيلز وبكره تنسوها وتستفرغوها في الامتحان as usual. It's not why I'm here, okay? I want you to learn something. If you care about understanding and potentially at some point helping a patient in the future when you practice medicine, then understand the concept. تعرفوش كيف ممكن تساعدوا patient في هذا الموضوع with things that you're trying to learn. And don't ever underestimate, basically knowing the necessary details. في actually quote by Albert Einstein who says, although he's not a role model to follow, basically. بحكي education is what remains after you forget everything you learned in school. So كمان مرة التفاصيل هي تحفظوها وبتروحوا to actually you put it in the exam and تنسوها, it's not really useful to you. المهم what remains after that. Okay? So what kind of understanding, what kind of, um, um, different changed individual you are in terms of how you can help people with your knowledge as a medical doctor. That's the purpose المفروض. Okay? So think about it this way. بناء على هذا الكلام كمان مرة اللي بينجح في الامتحان يحضر المحاضرة، يسمع المحاضرة، ياخذ نوتس. مش راح احاسبكم على اي شيء موجود في يعني I spent the past 15 years of my life studying neuroscience. These are like a second nature to me, and I'm not going to expect you to do that in a week, so مش عادل هذا الكلام. بس at the same time I expect you to put effort. على أقل تقدير تسمعوا المحاضرة كم مرة. لن يرسب إلى المصر على الرسوب. اللي بدوش they don't want to show their face, they don't want to study, then it's their problem not mine. Okay? فببساطة اللي بده ينجح يحضر المحاضرة، يسمع المحاضرة، يفهم الكونسبتس and believe me you will answer the questions. اللي بده يجيب 100 يقرأ التفاصيل، يقرأ الدراسات، يقرأ الشابترز. بلاش مشان ما تخافوش من الدراسات. اوكي؟ يقرأ الشابترز. Um, the recordings بحكي مع جيهان ان شاء الله just to make sure that you get at least the links لانه انا يعني زي ما حكيت لكم طلبت من جيهان من البدايه تبعت لي recordings عشان اساس to download the files to the folders بس ان شاء الله we'll get back to you today ان شاء الله. If not, I'll ask her to send you the links so that you can watch them. And meanwhile, um, let me answer this about the PNI. So yes, we do research في جامعة القدس, and we have been doing this for the past 15 years as well. Um, so anybody who's interested in neuroscience research, yes, you have an opportunity to do that at home. At Al Quds University, we have 6 research labs. Um, cognitive neuroscience, molecular neuroscience, neurogenetics, neurophysiology, and neuroendocrinology. We do a lot of work as well in terms of developmental disorders and developmental neuroscience and computational neuroscience. Actually, that's the core of what I do as well, and AI. So yes, if you're interested, you can basically look it up and potentially contact me. But basically, يعني, this is dedicated for people who want to pursue that as a path at this stage in their development, in their professional development. Of course, we always welcome people who want to come there as just quick volunteers and leave. بس انتوا شايفين الوضع الحالي للاسف ما بيسمحش يعني انه to actually kind of do any activities. ف المفروض انه من هون تهدى الاوضاع شوي. We'll be able to receive students at the PNI and actually you can actually contribute and participate in research. Keep it in mind. We'll potentially have people in the faculty inform you when we have the recruitment open, and then basically as such, get introduced to the research methodology. If you think I can be of help to you guys outside the PNI, don't hesitate to reach out to me, but with reasonable requests, okay? And I want you to know, and basically these are things I'll be more than happy to help any of you, but basically with things that I can do, all right? And I'll let you know. Okay, Dr. Hatim, yes, we'll give you the memory lecture. It's with him, and hopefully you'll have fun learning about memory from Dr. Hatim. And if there are any other lectures, CV, it's something that you need to بسأل عن CV. This is something that you can ask ChatGPT about. You don't need me, It doesn't take a PhD in neuroscience, basically in order to help write a CV. But basically that's really important, basically to consider. Maybe the person wasn't asking about writing a CV, asking about the application requirements. Yes, there will be prerequisites for you to apply to join the PNI, particular courses. لأن احنا we're very focused on the theoretical aspect of things لأنه للأسف فيش عندنا كثير capacity في البلد to do a lot of research with a lot of equipment. So we ask of you guys to know a little bit of Python, a little bit of statistics, a little bit of I mean, not to be expert, don't misunderstand, not to be software engineers. I'm not asking that. Well, as statisticians, but just the basics. You can run some, logical analysis. You can run some, design aspects. You can write properly, in English. اللي سألني a couple of days ago نحكي بالعربي نحكي بالعربي. Yes, we can. Nothing would make me happier. بس لغة العلم هي الإنجليزي. So you need to write properly, scientifically, in English, if you want to pursue that as not just a track in research, but also a track in being a clinician scientist if you want. This is actually something that is very appealing these days to people. So yes, I'll be more than happy to respond to questions. Maybe we can, anybody who's interested, you can actually reach out. There's the email neuroscience@alquds.edu. It's the email for the PNI. And you can look it up at neuroscience.alquds.edu is our web domain. It's under construction, has been under construction for a couple of years now. But basically we will be updating this. There's also, I think, a Facebook page if you want to follow it. And again, more than happy to be of help if I can help you. Okie doke. Any other questions? If that's it, then يعطيكم العافية ونلتقي في المحاضرة القادمة. السلام عليكم.